we move on to Dr. Guarner. He's already here online with us. Dr. Guarner, welcome once again, and thank you very much for joining us.
I'm very pleased for your kind invitation. So I'm going to share my screen and open the presentation, which I will follow up. I think the topic Dr. Ahmed Murad already mentioned, the importance or the relevance of the gut microbiome in digestive diseases. My role today is to cover the topic of the influence of the gut microbiome on immunity. And so the agenda includes first the brief description of the gut microbiome, then to speak about the role of the gut microbiome in adaptive immunity, microbes and adaptive immunity. Then the potential role of dysbiosis, or changes in the gut microbiota in association with chronic non-communicable diseases in modern society, like cancer or like inflammatory bowel diseases, like cardiac diseases, for instance. And then the potential impact of antibiotics in the development of dysbiosis and then in susceptibility to infections. So taking benefit from the case of COVID-19. I'm going to comment some data on COVID-19 that I think make easy to understand the importance of having robust and potent gut immunity in relationship with the gut microbiome. And therefore, I have a question for all of you. I would like to know your opinion about this. Which of these factors you think is the most relevant for developing a robust and balanced immunity on immune system? I know that all of these factors can be relevant, but please select only one. So cesarean birth, vaginal delivery, poor environmental hygiene, compliance with vaccination programs, a diverse gut microbiome, diverse richness in bacteria in the gut microbiome, frequent use of antibiotics, high rate of infections during childhood, natural genetics, so your own genetics, or in other words, inherited immunity. So please select one. At the end of this talk, we will review and comment your opinions. Then concepts to be mentioned. Genome. When we talk about genome, we are talking about the genetic material of an organism. Metagenome is the collective genomes of the constituents of an ecological community. So all the bacteria that are together, that play together, or all the plants in the forest that work together, they have a collective genome that's integrated in networks. Then microbiota is the community of microorganisms that occupy a well-defined habitat, like the gut. The gut microbiota will be the community in the gut. And the microbiome, the word microbiome and microbiota, initially microbiome was defined also the same as the microbiota, but now many authors, they say that it's more precise to consider the microbiome as the collective genomes of the microorganisms living in a well-defined habitat, somehow the metagenome of the microbiota. If we consider now the human body, in the first line, you will see here the number of cells in the human body. You will see that we have a lot of red blood cells, erythrocytes, and we have 38 trillions of bacteria, a lot of bacteria, and mainly most of them in the gut. But here we don't know about yeast or viruses. This is a lot of, numbers are even bigger. So we have a lot of different cells in our body that are non-human cells. But you look at the mass, of course, the mass is more important in terms of muscle cells, which is here, 20 kilograms of muscle cells in an adult human being, or adipocytes, so the fat tissue is also important. And in terms of mass, bacteria are not that important. Around 200 grams is what they estimate in this paper. We did study looking at the biomass in the colon of volunteers, healthy people. And then we found the biomass, which is not only bacteria, but also the fibers, degraded bacteria, and water, which is the medium they need to be alive. We found that before the meal, they would be around 700 mLs as an average. And after the meal, this would increase 1 or 2 hours after the meal, would increase by 200 mLs, 150 mLs. If the meal is high in fiber, the increase is higher. So there is more substrate going into the cecum and into the colon. And of course, after a stool movement, the amount of biomass goes down. So we were talking about 38 trillions of bacteria. We're talking about 800 mLs of biomass of living bacteria working inside our body. How many species? If you look to reviews in different scientific journals, you will see some journals, they say, we had 200 different species in our guts. Some of the journals we have they would say that humans have 1,000 or 2,000 species. Everything is true, because now the way we can check for different bacteria, this is diversity, means non-redundant bacteria, so different bacteria species in a fecal sample, depends on how deep you sequence, how many sequences you obtain from a given sample. You have the fecal sample, you extract the DNA, and you put the DNA in a sequence machine. And if the sequence machine obtains 1,000 sequences, then you will see up to 80 to 100 different species in that fecal sample. However, look at this study. This study was done with 2 million sequences, very deep sequencing, and then it would found up to 2,000 different species in a fecal samples, which means there are some dominant species, about 100 or 150 species that you can detect immediately when you are sequencing, but you go deep in the sequencing, you also will find these very rare and less abundant species that are in lower abundance in the gut. And interesting here, you will see that the diversity of species, OTUS means operating taxonomy units, is a way of nominating species by sequences. You will see that the Americans in the United States have less diversity than people in Malawi, in Africa, or people in the tribes in the Amazon River in Brazil. And genes, what is the human gut microbiome? How many genes we have inside our gut? This was done by the MetaHit project. I was part of the MetaHit project. There is an average of around 600 different genes that we have in the colon. And 300 of them is common to many individuals. In this cohort, we had individuals from Europe, but as well from China. So there are some genes that are present in almost every individual, and some of the genes are variable. And if you look at the genes, you can know which proteins are coded by those genes, and then you can learn about functions. This was the cohort of 125 individuals. And this means that some functions were only present in one individual, but some other functions are common. Again, about 6,000 functions are common to all individuals. And here in the blue columns, you will see which functions are more abundant in individuals in the gut microbiome, in the bacteria in the gut, which is the blue columns, and in yellow is those functions that are more common in the human genome. We have in our body, our human genes are 25,000, our bacterial genes or microbiota genes are 600,000. And you will see that our microbiome is specialized in metabolism. We have many, many metabolic functions provided by these bacteria that are very important to digest tomatoes or to digest aubergines or to digest nuts. We need those bacteria, whereas the human genome is more specialized in cellular processes. And the focus now is in immunity. if you look at the bowel wall of a rat, which a mouse, sorry, this is germ-free, has growing but had no contact with any microbes, you will see here the lymphoid follicles. The lymphoid follicles are inducers of immunity. The structures are there, but there's no activity there. Whereas if you look at the gut of a conventionalized mouse, which means a mouse that has a normal gut microbiota, you can detect by the arrows a lot of activity. So cellular proliferation, lymphocyte proliferation inside of these follicles, which means that the immune system is going on. In fact, this germ-free mouse do not have gamma globulins in serum, and they do have a lot of globulins. More in detail, this study was published in Nature some years after the other one, and this is the lymphoid follicles in a newborn mouse. You only see green and yellow and blue color. Green is this lymphocyte-induced cells, and the blue color is the dendritic cells. This is the antigen-presenting cells. So these cells capturates the antigen and present to these immature cells, and then they become lymphocytes, B cells. The B cells, the red color, these are cells that produce antibodies. And these antibodies go also to the serum. Then you have gamma globulins in the serum. And this, from birth, this moves to maturity. And you can see here that if you treat the mouse with non-absorbable antibiotics like vancomycin and colistin, they do not develop, they have a low number of these lymphoid follicles, these mature lymphoid follicles, both in the ileum as well in the colon. And in germ-free mouse, in fact, you don't have these follicles. They only develop in this SPF, means specific pathogen-free mouse. This is important because pathogen-free, these mice, there was no disease, there was no pathogen, but the immune system develops. You're getting antibodies against many things because your immune system develops by the interaction with the gut microbes. We're talking about mouse, but then we go to see what happens in the human being. Well, the human being has these lymphoid follicles as well. And these lymphoid follicles are situated in the distal ileum. At birth, we have around 100 paired patches. The paired patches are aggregations of 5 to 6 lymphoid follicles. And in a young adult, you will see 250 of these lymphoid follicles. And you can see here also the activity of proliferation of lymph cells. In the large bubble, we have about 30,000 lymphoid follicles. This is the inducers of the adaptive immunity. And how this works? This works because the antigen-presented cells, they capturate antigens from the lumen of the gut. And because of recognition with patterns in the surface of the antigen-presented cells that interact with the antigen, with the foreign antigen, they produce cytokines that modulate the maturation of naive T cells to develop as Th1 or Th17 that to very aggressive against the antigen, or Th2 that develop a lot of humoral defense against the antigen, or, and which is very important, if the antigen is recognized as friendly, the cytokines that are produced modulate this lymphocyte to proliferate as Tregs, which is the recognition of tolerance. So if this antigen induces tolerance, there is no inflammation in response to this antigen. This is very important because we need to recognize all the friendly antigens in our body, in the skin, in the eyes, wherever. So it's very important not only to reject pathogens, but particularly important to recognize friends and not to be inflamed against friends, because inflammation damages our tissues. This is one of the important issues. Is this relevant for humans? I tell you that I'm only putting one experiment, one evidence, which I think is very important. As you know, melanoma, when metastatic melanoma cannot be treated by chemotherapy because the tumor does not respond. We are using immunotherapy. Immunotherapy is a way in which the drug stimulates lymphocytes against the melanoma. So it's the immune system of the patient who destroys the melanoma. It's not the chemotherapy, it's the immune system. We stimulate the lymphocytes. And it's interesting, here you can see the survival of the progression free of melanoma in metastatic melanoma patients after treatment with immunotherapy, anti-PD-L1. And you will see that people with high diversity in fecal samples, that's better. They do better. These people with high diversity, they do much better in terms of survival. Very, very curious. And what they did is that they had fecal samples from responders in blue color or non-responders in red color, and they had germ-free animals. There was transplantation of feces in the germ-free animal, and then the feces, the microbes in the feces, they were instructing the immune system of the animal, of the mouse. Then the melanoma was implanted, and then the immunotherapy was started, which is the anti-programmed cell death L1, which is avoiding apoptosis of lymphocytes. And I think this is really very interesting. You see that those mice who were treated with feces of non-responders, the immune system was weak and immunotherapy was not efficient. And you can see here the volume of the tumor was growing. There was no effect by immunotherapy. Whereas those who were transplanted with feces of the responder, they had a more robust immune system. The melanoma was implanted, the immunotherapy was started, and when starting the immunotherapy, the tumor did not grow anymore. And this was related with a lot of lymphocyte cells killing the tumor. So this is really relevant for humans. And the other side, this is again immunotherapy in patients with lung cancer or with renal carcinoma. And this is also months of survival after immunotherapy started on month 0. This is a month of survival. And the red line is people or patients who had treatment with antibiotics for any reason during therapy during immunotherapy. And you can see that the survival was worse. So giving antibiotics to people on immunotherapy reduces diversity of the microbiome, and then the response to the immunotherapy, the immune system weakens, and the response is not as good. And the same was proven in another hospital. This was done in France. It's in England. And they found also that prior antibiotic therapy, before the immune checkpoint inhibitor, this is immune therapy in patients, again, with melanoma or lung cancer and other similar types, this was associated with worse overall survival, 2 versus 26 months. So what happens with antibiotics? This is a study published only 4 days of antibiotics. You can see the drop here in richness of bacteria in fecal samples or in Shannon diversity, which is another index. And day 8, so 4 days after antibiotic therapy, the recovery is not coming. Only after many days you have a full recovery. You can see here the plasticity between a bacteria in the gut and the immune system. Even in older people, 60 years old people that go for immunotherapy because of a carcinoma, then just because having antibiotics reducing diversity, the immune system is not as potent as it used to be. The other thing is that the loss of microbial richness is a common feature of chronic non-communicable diseases of modern society. This means that many of these diseases, like metabolic syndrome, obesity, type 2 diabetes, inflammatory bowel diseases, also some functional bowel disorders, Risk of type 1 diabetes and celiac diseases in children has been shown before developing the disease. They have a drop in diversity. And also colorectal cancer, breast cancer, and also certain mental and neurodevelopmental conditions such as autism and spectrum disorder. It has been found they have lower risks. All of these diseases, they are of increasing incidence in modern society. These are data of the world. It's not rich countries, all countries. You can see here in red color, death because of infectious diseases or maternal neonatal diseases. It's about 10 million deaths in 2017. Look now, the blue. The blue is death because of non-communicable diseases, 41 millions. This is cancer, this is diabetes type 2, obesity, cardiovascular disease. This is not transmitted by any. This is really related with a bad lifestyle. And we see that in all these diseases, there is a defect in the gut microbiota. You know, this year, if we have 1 million deaths more by COVID-19, still the killer is cancer. The killer is type 2 diabetes. I think this is important because in our practice, we will have each time more concern, not about H. pylori, Helicobacter pylori, but about IBD, IBS, colon cancer, as you all know. And the hypothesis, and this is an interesting paper published in Science before the COVID pandemic, they were saying that the microbiota in farmers or in hunter and gatherers, so in tribes that they live in the very old lifestyles, living far away from the industrialized society, the microbiome would be here. Here is Venezuela and Burkina Faso, Peru, some data from different countries. And here at the end, you will see data from Ireland, Canada, United States, Australia. So this is the microbiota has changed a lot. And these changes came in 100,000 years from the hunter-gatherers, maybe 2,000 years farmers, but the Industrial Revolution is only 100 years, 150 years, big changes in the microbiota. Our genome has not been adapted. And because of the changes in diet, the changes in the antibiotic use, not breastfeeding, so the microbiota is different. And they conclude, I think it's in this review, if you want to, it's free, you can download it. So they say microbiota alteration and the accompanying loss of certain functional attributes might result in the microbial community of people living in industrialized society being suboptimal for human health. This is the concern now, that probably we're losing capacity in our microbiota to regulate immunity, to properly regulate immunity, and this is related with obesity and with many different diseases. The case of COVID, if you look at the map of May 20, you will see that it's Europe and the United States where most cases accumulate. So in industrialized society, the first wave affected very much to the industrialized society. But even if you look at mortality, now at the date of October 7th, so it was yesterday, data retrieved yesterday, you can see that mortality, which is the confirmed deaths from COVID-19 per million people in the different populations, you will see Spain, United States, United Kingdom, Italy, France, and Netherlands on the top. And you can see here Saudi Arabia, Qatar, Lebanon, Egypt, with much. So the disease came later, that's right. But look at the steep slope. From when the virus was there, immediately started fatalities, death. Here is not that steep slope. The virus was already in these countries, but it's not, the mortality is not growing. So there is a resistance to mortality. And you would expect that in countries with high income, with very high domestic product, with richness. They would have better national security, better hospital, more ventilators, more facilities, intensive care units, but they have more mortality. This is a logarithmic scale. Rich countries have higher mortality than other countries. You can see here Egypt, Lebanon, Tunisia, Jordan. Many. Is why if the health facilities are here is less mortality here. There are many reasons. It's a lot of discussion on this. I think it's an interesting discussion. But one of the things is that, of course, in rich countries, in high-income countries, life expectancy is higher. But the difference is not that much. The difference is not that much. And the other thing is that the use of antibiotics, so the daily dosage of antibiotics per 1,000 population, data from 2015, is much lower in low-income countries. It's contrasting because these countries have more infectious diseases, but they are using less antibiotics than the high-income country with the incidence of infectious diseases much lower. So, and this is almost double. This is almost double, which means the gut microbiome in these countries is worse, as suggested or proposed by the science paper, the vulnerability paper. So, but again, it's true that most of the mortality in Europe came from people above 70 years of age. This is data from South Korea, Spain, China, Italy. Mortality is related to the elderly. And this is because in the elderly, they have immunosenescence. Immunosenescence means that they don't have the good facility to react to an antigen, but they have inflammation. And this was even worse in people that were resident in long-term care facilities. So people at home were more protected than people in long-term. Almost half of the people dying in Europe were living in long-term as in April 2020. Why? Because we don't know, of course, but I think there are some interesting data here. Here is a study that was done in Ireland in 2012. And you will see here the levels of different cytokines, inflammatory cytokines being higher in levels in people in long-term facilities, people in care, elderly living in care facilities, not at home. Green color is people living in the community. So you can see the C-reactive protein is higher, TNF is higher, IL-10 is not higher, but this is the anti-inflammatory cytokine. IL-8 is higher, IL-6 is higher. And together with this, you see phylogenetic diversity in fecal samples. So these fecal samples from these people had lower diversity. You hear it's around 100, more than 100 dominant species per sample versus in long stay below 70 species per sample. And this was also related with food. People, they did a score for diversity on healthy food, which means diversity on vegetables, fruits and vegetables. People in long stay had lower intake of these healthy foods or less diversity, and this was also related. So less healthy food, less diversity, and more inflammatory component. Death is because the lungs get inflamed. The pneumonitis, you know, that if the COVID patients, when they get a pneumonia, if they have an excessive inflammation, they have this cytokine storm, cytokines being up and destroying the lung, destroying the alveoli, and then these patients they die by sepsis, even if they have. So the other thing what happens in nursing homes is that the use of antibiotics is really very high. This is some data from Canada, where the index of DDD, this daily defined dose per 1,000 inhabitants per day, is 17 in the community, but in nursing homes goes from 25 to more than 120 in the different facilities. So the use of antibiotics again. And now finally, we're going to look up the data in COVID patients. This is the microbiota in healthy subjects, this blue color. And this is the microbiota in COVID patients without antibiotic treatment. So it's a change, but not that much. Whereas those patients with COVID treated with antibiotic changes are much more important. And what is very clear is that there is a positive correlation between severity of the disease and the presence of bacteria, and a negative correlation and the presence of Bifidobacterium anaerostypic. This is a butyrate producer, Faecalibacterium roseburia. These bacteria are famous because these bacteria are low in Crohn's disease. These bacteria are low in cancer, in colon cancer. These are the butyrate producers. So the lower these bacteria, the lower, the worse the evolution of the disease. So we are now getting to the end of this talk, and I would like to ask you again for your opinion. Please, if you could answer to this question, what's your opinion about the most relevant factor for developing a robust and balanced immune system? Cesarean birth, vaginal delivery, poor environmental hygiene, compliance with vaccination, a diverse gut microbiome, frequent use of antibiotics, high rate of infectious disease in childhood, natural genetics. I don't know, you have some, I think that after, we will see now the results of the two polls, what were your opinion first, and what is now your opinion. And I think they will move to the next slide. I don't know if Valentina will put the slide with the results when ready. Yeah, I guess, or if not, I can go to, yeah. Okay, here we have interesting. Here we have the. So the winner is natural genetics, people with natural genetics, but I was saying about developing a robust and balanced immune system. So the growth, but anyway, natural genetic was the first, and then came frequent use of antibiotics, and then a diverse gut microbiome. And then can we see the data in the second survey, please? If we can look at that. All right. Yeah, thank you. Thank you so much. Now, many people share the same opinion as I have. It's my opinion as well. I believe that having a diverse gut microbiome is something that we don't have in mind, but probably is important in order to develop. Maybe natural genetics is very important, but we are talking about the adaptation, the immunity that develops during life, over the life period. So thank you so much for your participation. Now we go to the take-home messages, which is my last slide, and I will move to the last one, which is the take-home messages. So the gut microbes induce adaptive response via gut-associated lymphoid tissue, the GALT, and regulate local and systemic immunity, because all the antibodies, they spread all over the body. Diversity matters. A robust and regulated immune system is usually associated to high gut microbial richness. This biosis of the gut microbiome has been incriminated in the origin of inflammatory, metabolic, or neoplastic disorders of increasing prevalence in modern society. So it's the risk factor that is coming, this biosis. Therefore, we need to develop our mind in terms that the target now is maintaining gut microbiota diversity as a novel clinical target for health promotion and disease prevention. And probably this is done by healthy food and careful use of antibiotics. Thank you so much.
Thank you, Dr. Guarner. A very up-to-date presentation. I think very relevant in many ways, and mentioning this word that all concerns us at the moment, no matter where we are in the world, COVID-19. And I think in the context of gastroenterology, also a very relevant topic. So thank you very much for that. I would now like to use the next minutes here asking you to answer some of the questions that we had.
Thank you.
You mentioned, of course, now that the microbiome is important, and convinced, as it seems, many people that this is the key factor. Now, in the end, you said, okay, you already gave some hints now how to do it. No? And I think maybe here we have a lot of questions asking, what role do probiotics play in this still? I mean, how effective do you think are probiotics? What impact does meat have? Right.
Okay, I think that, yeah, probiotics might be useful. For instance, if you need to use antibiotics, it's better to use associated probiotic. But to be, in order to have a robust or diverse microbiota, it's not probiotics. Probiotics are helpful in different situations. I've used probiotics many times, but it's much more important healthy food, healthy diversity. I couldn't show. There are now many data. There was a study in the United States, but they were thinking that vegans would have better microbiota than omnivores. People, it's not true. What made a difference? The difference was made in people with a diverse diversity in vegetables in food. People eating more than 30 vegetables or fruit per week have better microbiota in terms of diversity than people eating less than 10 vegetables or fruits per week. Even if they were vegans or vegetarians in a very monotonous diet, the diversity in the microbiota was low.
Okay, interesting. There's a couple of questions that also obviously all around the microbiome now. Can charcoal protect the microbiome from antibiotics?
I think this is an interesting thing. I don't know. That's it. But I think it's important to check. I believe one of the things important for the gut microbiota is the anaerobic situation. So because those bacteria that are no good are the aerobes. The aerobes can invade the mucosa, can inflame, and the aerobes provoke sepsis. And in fact, COVID patients, they die by sepsis and multi-organ failure. Is the translocation of bacteria. So if you have an anaerobic environment in the colon, which is very important, then you have many of these Faecalibacterium, Roseburia, which are they are obligate anaerobes. And it might be that charcoal helps. I don't know the data but I think one of the results is that it captures gas and particularly oxygen. So, right.
Okay. Now, what do you think is the best management option for improving the gut microbiota diversity? I think is then still the questions that's out there, and maybe couple that with a question coming in here through our Q&A is, how long does it usually take to get to that state, to the desired state of diversity once you initiate changes?
Right, okay. So this is something that is being studied very much. So they have seen in very drastic changes, so very important changes in the diet from day to day. Even in 7 days or 2 weeks, there is a study in South Africa with 2 weeks where they change also the inflammatory markers in the colonic mucosa, which is very interesting. But then, diversity does not increase in such a short period. But you can get more butyrate producers. So it seems that you need to have longer periods, more than 1 month, 3 months, in order to get diversity, and try to incorporate all kinds of vegetables. I'm not talking about salad. I'm not talking about only apple. I'm talking about aubergines, peppers, onions, tomato, everything. It's not only green, but red color or multicolor.
Okay. Here I have a question that actually I've seen now a couple of times as well. Is a fecal transport an option for treating, for improving the microbiome as one, but also for treating COVID-19 patients potentially, and other diseases or symptoms? states such as obesity,
Right.
liver disease?
A fecal transplant is very successful in one particular situation, which is recurrent diarrhea by C. difficile, which is a bacterium that in people that has a lot of times received antibiotic therapy, the diversity of bacteria in the gut go down. And then there is an overgrowth of this Clostridium difficile that produces a toxin, and they have many diarrheas. Then the only way is to do a fecal transplant. But besides this situation, efficacy is not as good. So there was a study in type 2 diabetes. By fecal transplant, this was an improvement, but was transitory. Because at the end, if you don't feed your bacteria, you will lose the bacteria introduced by the fecal transplant. So it's an ecosystem. And yeah, you can put foreign bacteria inside. It can work for a while, but if it's not the adequate environment, we don't know how, but we are considering diets, but maybe there are things. Over a period of 2-3 months, the effect disappears.
Okay, we have here the question always relating to some drugs. It's not difficult for me maybe to pick them all out, so metronidazole is mentioned, proton pump inhibitors are mentioned.
Right.
And what do you think is their effect, and maybe other drugs that come to mind because you
right. Okay.
What effect do they have on the microbiome?
Yeah. Shortly, metformin, that's for type 1 diabetes, has changes in the gut microbiota that are beneficial. So it has been that metformin improves the butyrate producer. So metformin has changes beneficial. Probably is the only drug that clearly. All the antibiotics have detrimental effects. All of them reduce the butyrate producers. So antibiotics should only be used in cases where you have cleared the benefit. When you see the benefit, you need the antibiotic for a dental, but whatever you have to use for diverticulitis, whatever. But they're always wrong. And there are already observational studies with children where children below 10 years of age, if they receive several times antibiotic during childhood, they have higher incidence or higher risk of Crohn's disease, ulcerative colitis, obesity, and colon cancer as well. There are studies on colon cancer and polyps. So they have more, not cancer, but more polyps related. So it's not only the dysbiosis that is generated for a while, but also consequences in the long term, particularly during childhood. Proton pump inhibitors, they do changes, but we don't know if these changes have a relevant health outcome. With antibiotics, we know that they have a negative health outcome for the unnecessary or excessive use of antibiotics. Proton pump inhibitors, yes, they change the microbiota, but there is no clear, there is no increased risk of Crohn's disease, there is no increase of obesity. We don't have data so far.
Okay. Here I have a follow-up question to the fecal transplant, asking what is your criteria for the donor of a fecal transplant? What are your criteria for using a fecal transplant?
Right, yeah. I recommend to download from, because it's free, from Gut, the European journal called Gut. There is a consensus for the criteria for a fecal transplant. This is already 2 years old, but there are all the criteria on the selections, which is complex, the selection of the donors. And a novel thing is that also you have to cultivate this fecal sample of the donor with antibiotics, because if you found multi-resistant bacteria, if something is growing in the middle of many antibiotics, you cannot use that donor. So this was not in the first consensus, because the experience in the United States was some deaths because multi-resistant bacteria in feces were transplanted to individuals. And then when they sick, these bacteria was getting an infection or diverticulitis, whatever, and there was no therapy to treat them, and they would die. So they were at least to death because of that. So there is a very well described protocol for selecting the donor and for testing all the things in the donor in order to be on the safe side.
Okay, maybe finish up with a final question and then we move on. Is here dairy? How do you see dairy? I mean, there's some people also that say, you know, milk is beneficial, others are critical. How do you see the effect of dairy on the microbiome?
So yogurts, fermented foods are interesting in terms of that not only yogurts, kefir, whatever, the fermented product, and also other fermented products, because our guts needs, we believe, this is still on discussion, but a lot of people believe that the gut has that many lymphoid follicles in order to receive bacteria every day. If you are in New York and you buy vegetables, they are in a plastic bag, and inside is a bacteriostatic atmosphere. Everything is clean, no bacteria. You don't eat any bacteria. They did a calculation, and as a person in New York, even if he or she is vegetarian, no bacteria are passing through the gut. And the idea is that the more bacteria, the more good bacteria passing through your gut, your immune system reacts better. So a sterile food is not the best way, unless there is an infection. So yeah, everything must be balanced. If it's tuberculosis at home, if it's an infectious disease, you need to keep all the hygiene. Hygiene is still very important, but it's no infection. All the good bacteria, the more the better.
Okay. Dr. Guarner, thank you so much. Thank you so much for joining us. It was a great pleasure.